In the previous lecture, I have introduced you single gene inheritance or Mendelian inheritance. In genetics, exceptions to Mendelian inheritance do occur. Some traits are passed on through non-traditional types of transmission. Today, I'm going to introduce you non-traditional inheritance, and we will cover 4 topics. First, we will look at mitochondria inheritance. Mitochondria DNA has 3 features. One is circular. Second one, the size is around 16.6 kb. Third, mitochondria DNA encodes 13 polypeptides. Proteins encoded by mitochondria DNA make up subunits of the electron transport chain. Therefore, proteins in the electron transport chains are encoded by one of the 2 genomes, the mitochondria and the nuclear genomes. Mitochondria DNA does not follow Mendelian inheritance because paternal mitochondria are normally not transferred to the oocyte during fertilization. Therefore, diseases caused by mutation in the mitochondria DNA are maternally inherited. As shown in this pedigree, a trait is passed on only through women. All or almost all offspring inherit the trait. Another feature of mitochondria DNA is that it has a higher rate of spontaneous mutation than nuclear DNA. Here are some mitochondria genetic disorders. We have covered them before. Here is integration with electron transport chain. For example, mutation in NADH dehydrogenase decrease the activity of electron transport chain and oxidative phosphorylation and reduce the production of ATP. These mitochondria disorders are summarized in this table. Heteroplasmy refers to presence of more than one type of mitochondria in the cell. Here is a cell. These are mitochondria. Mutant mitochondria are shaded. During cell division, mitochondria separate passively. By chance, some cells receive more mutant mitochondria. Other cells receive fewer mutant mitochondria. Cells with more mutant mitochondria have a stronger phenotype. For example, child from the egg containing 20% of mutant mitochondria does not have any phenotype. A child from the egg containing 50% of mutant mitochondria has mild phenotype. A child from the egg containing 80% of mutant mitochondria has severe phenotype. Heteroplasmy can explain what is seen in this pedigree. This is a pedigree of Leber hereditary optic neuropathy. The mother, one daughter, and one son do not show any phenotype, but the rest 3 children do. Heteroplasmy can also explain why the son of this woman doesn't show any phenotype. Threshold effect refers to sudden and radical change in a phenomenon, which often occurs after passing a quantitative limit, which is called the threshold. Clinical expression of a mitochondrial DNA mutation is determined by a relative proportion of mutant and wild-type mitochondrial DNA. Small percentage of normal mitochondrial DNA may be enough to maintain cell function, but the rate of ATP consumption determines the vulnerability to mitochondrial mutations. Tissues affected mitochondrial mutation most include central nervous system, heart and skeletal muscle, renal, endocrine, and the liver.
Now, let's move to part 2. We will look at trinucleotide repeat expansion.
In a normal individual, CAG is repeated around 5 to 35 times. But in a disease condition, these three nucleotides are repeated 30 to 300 times. For another example, in a normal individual, CGG is repeated 6 to 50 times. But in a disease condition, CGG is repeated 50 to 1,000 times. CAG tends to be repeated within a coding region. CAG repeat expands in size in Huntington's disease and few other diseases. CTG repeat expansion occurs in 3' UTR in myotonic dystrophy. CGG repeat expansion occurs in 5' UTR in Flageolet XA mental retardation, or in an intron in Flageolet XE mental retardation. This table shows genes affected and the related disease. CGG repeat affects two genes, FMR1 and FMR2. Both are located on the X chromosome. CAG repeat affects multiple genes. For example, androgen receptor, huntingtin, ataxin-1, and atrophin. CTG repeat affects muscle protein kinase, and GAA repeat affects frataxin gene. When repeats occur in a protein coding region, they make the protein toxic. When repeats occur in RNA coding regions, they alter RNA function. When repeats occurred in non-coding regions, they reduce transcription or translation. Huntington's disease is caused by CAG repeats in exon 1 of the huntingtin gene. People with Huntington's disease have 39 or more CAG repeats. People with Huntington's disease show jerky, random, and uncontrollable movements called chorea. Their lifespan, on average, is 15 years after the onset of symptoms. Flageolet X syndrome is caused by expansion of CGG repeats in the 5' UTR of FMR1 gene. CGG repeat expansion occurs in female meiosis. Flageolet X syndrome has traditionally been considered an X-linked dominant condition with variable expressivity and possibly reduced penetrance. However, due to genetic anticipation and X inactivation in females, the inheritance of Flageolet X syndrome does not follow the usual pattern of X-linked dominant inheritance. People with Flageolet X syndrome have prominent forehead, large jaw, and large ears. Affected boys have large testes. They have mental retardation of varying degree. Myotonic dystrophy is an autosomal dominant disorder. People with this condition have muscle wasting and muscle weakness. Myotonic dystrophy is caused by CTG repeats in the 3' UTR of DMPK gene. DMPK encodes myotonic dystrophy protein kinase. Friedreich ataxia is caused by expansion of GAA repeats in the first intron of the FFN gene. This gene encodes frataxin, an iron-binding protein in mitochondria. People with this condition have muscle weakness and loss of coordination. Here is a table to help you remember trinucleotide expansion and the corresponding disease. Here, U is pronounced as A, similar to pronunciation of A in words such as task and ask. Comparison of these 4 diseases are summarized in this slide. In some autosomal dominant disorders, such as myotonic dystrophy and Huntington's disease, the onset of disease occurs at an earlier age in the offspring than in the parents, or the disease occurs with increasing severity in subsequent generations. This phenomenon is called anticipation. Here is an example of anticipation. This is a pedigree for myotonic dystrophy. This man carries 5 CTG repeats on one chromosome and 65 repeats on the other. The number of CTG repeats is increased to 90 and 95 in his 2 children. The number of CTG repeats is further increased to 150 and 200 in his 2 grandchildren. This slide shows how the trinucleotide expansion occurs. During meiosis, one chromatid incorrectly pairs with the corresponding sister chromatid. Unequal crossover leads to one chromosome gaining DNA and the other chromosome losing DNA. The one gaining DNA produces trinucleotide repeat expansion.
Now, let's move to part 3. We will look at mosaicism.
Mosaicism refers to the phenomenon that a person consists of more than one genotype. Mosaicism can be somatic or germline, depending on when the mutation arises in development. If the mutation occurs early enough and it is present in all or some of germline cells, it will be transmitted to the offspring. In some families with autosomal dominant disorder or X-linked recessive disorder, the parents are phenotypically normal. It turns out one of the parents has germline mosaicism. It means the mutation is present in a proportion of the gonadal cells of that parent. It is important to keep germline mosaicism in mind when providing recurrence risk in genetic counseling for apparently new autosomal dominant or X-linked recessive mutations. For example, in this pedigree, the mother of the 2 affected children is a mosaic for the mutation. She does not show signs of the disorder. If she is a germline mosaic, only X cells would carry the mutation. Very similarly, this pedigree also suggests a germline mutation in individual of 11 or 12.
Now, let's move to part 4. We will look at genomic imprinting.
We now know that some genes from the father or mother are expressed differently. The parent of origin effects is referred to as genomic imprinting, and methylation of DNA is the main mechanism by which expression is modified. Evidence of genomic imprinting has been observed in two well-known disorders, Prader-Willi and Angelman syndromes. In somatic cells, some regions of chromosomes are methylated differently on paternal and maternal chromosomes. But in the primordial germ cells, methylation pattern is reset. The residing gametes adopt only one type of methylation pattern, either paternal or maternal. That imprint will stay on in zygotes. This provides a mechanism for parent of origin effect. Some individuals have inherited both homologs of a chromosome pair from only one of their parents. This is called uniparental disomy, as shown here. The conceptuals would originally be trisomic, with early loss of a chromosome, leading to disomic state. If an individual inherits two copies of the same homolog from one parent through an error in meiosis II, as shown here, then this is called uniparental isodisomy. If the individual inherits the two different homologs from one parent through an error in meiosis I, and this is called uniparental heterodisomy. Using DNA techniques, uniparental disomy has been shown to be the cause of a father with hemophilia having an affected son, and of a child with cystic fibrosis being born to a couple in which only the mother was a carrier. The majority of patients with Prader-Willi syndrome have a small deletion on paternal chromosome 15. Around 25% of individuals with Prader-Willi syndrome have maternal uniparental disomy. Babies with Prader-Willi syndrome have a hyperphagia, obesity, short stature, hypogonadism, and learning difficulty. Opposite to Prader-Willi syndrome, the majority of patients with Angelman syndrome have a deletion on maternal chromosome 15. A small number of cases arise through paternal uniparental disomy. Babies with Angelman syndrome have epilepsy, unsteady gait, severe learning difficulties, and a happy facial expression, as shown here. Here is a comparison between Prader-Willi syndrome and Angelman syndrome. This is paternal chromosome 15, and this is maternal chromosome. They are methylated differently. The majority cases are caused by deletion either on paternal chromosome for Prader-Willi syndrome or maternal chromosome for Angelman syndrome. Around 20% of cases are caused by uniparental disomy. A very small number of cases are caused by imprinting error.